Hello, we start today the second lecture in BIOL121. We finished the last lecture about with the characteristics of the living organism. So, in the second lecture, we'll talk about the domains of life. So we have like different domains of life in the classification. We will not deal with the classification in specific or in details. So we just mention the major domains of life. So we have the first domain is the bacteria, which is the most diverse and widespread spread prokaryotes. And there is multiple kingdoms under this domain. And you can see from the figure that each row, the shade structure represents one bacterial cell. While the other domain is the archaea domain, and the archaea means very primitive. And this domain have recently been separated from the bacteria domain because it possesses a very special characteristics, like the ability to live in very harsh and extreme environments, like salty lakes and boiling hot springs. This domain contains as well multiple kingdoms, and you can see each round structure in the figure represent an organism. The third domain is the domain eukarya, and it consists of a number of kingdoms. We'll focus about the kingdoms, not in details, like the kingdom planta, and all of us know the plants. And it is a multicellular terrestrial, multicellular eukaryotes that carry out photosynthesis and can make food by its own self. So it is autotrophic through the conversion of light energy into chemical energy. While the second kingdom is kingdom fungi, and actually is named fungi based on the mode of feeding of its members, like the mushroom. So they absorb nutrients from the outside their body. So they don't have the capacity to do the food inside their body. So they just secrete enzymes over the substrate and digest it and then absorb it from the surroundings. Then the domain Animalia, which we as humans belong to that kingdom Animalia, which we as a human belong to this kingdom. And of course, it consists of a lot of multicellular eukaryotes that ingest other organisms. So actually, it's heterotrophic. The last kingdom is protists, and these actually are unicellular and some relatively simple multicellular relatives in this kingdom. And these protists actually have a lot of characteristics in common between other kingdom, and still in debate to classify protists in a way to reflect actually its evolutionary relationship. So these are the main domains of life. So you need to know that all living organisms have some characteristics in common. Like all of them have a cytoplasm, a cell membrane that surrounds the organism, the cells, and the DNA as a genetic material. Of course, all of them have ribosomes in the cytoplasm. And ribosomes actually it is the machinery used for protein synthesis. You can see here a major difference between animal and the plants. You can notice that the major difference appear in the mode of feeding. Animal are heterotrophs while plants are autotrophs. And as well in the cell walls, plants are characterized by the presence of a cell wall that is made of cellulose, and as well contain chloroplast, which is not present in the animal cells, and as well animal cells have no cell walls. So you can see here generally how animal is classified. You can see it is classified according to increased in similarity from kingdom up till species, as well as decreased in similarities up from species to kingdom. And you can see how the group of animals are collected in a domain. You can see a lot of variety of animal that soon comes in classification until you find the species. And usually the species have a binomial nomenclature, consists of a genus names like Urus americanus. This pair belong to a bigger group, and the bigger group, which is the order, families, and blah, 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 etc., until reaching to the domain, the big domain, which is the eukarya. So this slide represents like how animals are classified and then subdivided until reaching to the species, according to, of course, according to complexity and similarities. We'll talk, if we are not familiar, Charles Darwin, who first wrote his book in 1859 about the origin of species, he studied the evolution in species, and he had a lot of theories concerning the developmental evolution of animals and how they try to adapt to the surrounding environment to survive. And its famous theory is the natural selection. That means that the nature select the fittest organism to survive. And in order to select the fittest organism to survive, this organism need to adapt. And in the following slides, following few slides, we'll see some of the adaptation of these animals. So let's look here why these ants have very long legs that is not proportional to its body, and they are slit-like legs, and these legs help this small organism, small animal, to survive in the desert heat. Actually, these long slit legs gives the animal very advantage and very adaptation to its surrounding, the desert environment, which is characterized by extreme heat. The elevated leg, these long legs elevate this ant almost 4 millimeters above the sand. And by elevating the body of the ant, it will have 6 degrees Celsius lower than the ground level. So these long legs help the ant to avoid the extreme heat of the ground. As well, these long legs help this ant to run as fast as 1 meter per second. And this as well help the ant to avoid the heat. You can see as well, it can use in the bottom left figure, you can see as well that the ant, these long legs help the ant to feed over a larvae. You can see that it can stand of the larvae and start to eat. So this is a kind of adaptation that happened over years to adapt this ant to live in a very extreme condition like the desert. So what are these ants doing? You can see that in this figure, the ant is like pushing liquid from its abdomen, while this ant as well doing the same, pushing liquid from their abdomen. While this beetle, you can see from the abdomen, it sprays around like some sort of liquid. So actually this wood ant, when threatened, the ant shoots volleys of formic acid. And this formic acid, they shoot it from their environment, their abdomen to the environment to defend themselves. While the beetle actually sprays a boiling hot liquid containing irritating chemical, and as well it is used as a defense mechanism against its enemies. So in all these cases, it's a kind of adaptation to survive. Here as well, you can see how the mating between 2 different bears and the hybridization between this bear and this bear can result to a new bear. And in this case, a lot of these happen in the nature for generating a new characteristics, since the genes of both pairs are mixed to give rise to a new individual with different genes and different genetic background from the parents. And it can survive, of course, it can survive more in the environment. And you can see how the polar pair mixes the genes from both parents to survive in the on ice with the extreme cold conditions. This is a perfect example of adaptation to environment. You can see this is actually the same fish. This fish is called mosquito fish. So mosquito fish, if this fish exists in a lake with high predators, this fish over evolution tended to have a very strong, powerful tail. Very strong, powerful tail. This enables the fish to rapid speed of swimming inside the lake. while to escape, of course, to escape enemies or predator. While the same fish in another lake with there is a few or low predation, you can see that the fish have like not a very powerful fin, back fin, and this to enable it for steady swimming. So actually, that's why the environment, or that's how the environment control the characteristics that the organism have, and it enables it to survive and adapt. In other words, in the first case, if the fish fails to have this powerful fin at the back, or the tail fin, and it fails to have, it failed to adapt, it failed to modify this, the other fish which failed to adapt and modify will die, or got eaten by the predator. So actually that's a reflection to how nature select the fit organism to survive. As well, you can see here that some organisms, some animals have evolution in their animal size and shape. You can see all of these three animals, seal and penguin and tuna, have something called convergent evolution. What's the convergent evolution? Convergent evolution, that means that different animals mainly aim to have, in this example, mainly aim to have an adaptation that enables this organism to be a fast swimmer. So they all have in their evolution conversion to a certain point to be a fast swimmer. So basically each animal try to adapt in a way to have like a fusiform shape of the body, tapered, which is tapered on both ends, that enables the animal to move faster. Actually, this you can see in the tuna or the ray fin fish, you can see it can swim up to 80 kilometers per hour, okay? And of course, this is helped by its fusiform shape. Here you can see the difference between horse and dog. You can see that both of them must have a common characteristics, which is the presence of hair. If you know that hair on our body or any animal body, it's a very characteristic of as a mammal feature. So if you saw any animal or any organism that have hair on its skin, you don't need to look for a classification for that animal. You are sure now that this animal is a mammal, because hair, the presence of hair is very characteristic to mammals. So you can see here that these two groups of animals are classified into horse-like and dog-like mammal. And you can look for both figures and you can see features. What do you think the feature from these two figures that may differentiate both of them? By other means, how can you say that this is a dog-like mammal and this is a horse-like mammal? Can you tell me at least the two features that is different between both two figures? خالد اتفضل يا خالد، انت رافع ايدك.
استاذ كان دلك انه شكل النوز تبعهم، انه اذا هو كان رفيع وطويل او اذا كان اعرض شوي.
That's good. ممتاز يا خالد. في حاجة تانية، في فرق تاني. Can you see another difference between horse-like and dog-like?
طول الدينان.
طول
الايرز.
شكل الدينان هو قصده.
ears, of course, ears. Ears are longer in dog-like than horse-like. Any other difference?
الهاي الطول.
الهير على النك بتاع الهورس.
الهير على النك في الهورس-like، صح. Any other difference?
الهايت.
الهايت of
size of the head.
It could be. خلاص, but there is another obvious difference.
الcolor.
tail.
لا الكالر لا طبعا.
tail
شكل التم.
nose
الماوس it's important different the mouse as well. Yes.
دكتور nose
النوز already said خالد already said the nose. حد عنده حاجة تانية؟
The hair.
Already said the hair.
They eat. That's it.
Okay. The neck.
size الحجم
The neck.
What they
Okay.
eat.
Have you noticed the neck?
yes
Which is longer, horse-like or dog-like?
horse like
Horse-like. خلاص?
The horse.
The horse. Why do you think the shape of the mouse is different in horse-like than in dog-like?
فيك تعيد السؤال بعد عمرك
Sorry?
Repeat the question please
Why do you think that the mouse is different? The shape of the mouse is different from dog-like to horse-like.
شايف لهم خير
Sorry, again?
It's because the species developed this way for their consumption needs. For example, horse-like animals, their mouths and jaws are built differently so that they can eat different various types of plants. But dog-like species, their mouths are shaped in another way so that they can
Because
eat
they eat
different
meat.
meats.
Okay, actually you are saying that one of them is herbivorous and one of them is carnivorous. That's the simple answer.
I guess so. But
Okay.
حتى between different carnivores في different mouth shapes based on
Yeah. I'm just comparing between these two pictures now. Okay, so one of them is herbivorous and one of them is carnivorous. Okay, I'll tell you another trick to identify carnivorous and herbivorous animals as well. Another trick.
Canines.
Sorry?
النياب.
Sorry?
الcanines. النياب.
Of course, if its mouth is closed, you will not know if there is a canine or not.
Okay.
Right?
Right.
Okay. From the head, you can know if the animal. It's another trick. You can know if the animal is a carnivorous or a herbivorous, or in other way, you can know if the animal is a predator or a prey. Okay, how that? By the position of the eye. If the eye is located on the sides of the head, that's a herbivorous. Majorly, 90% will be herbivorous animal. Or if the eye is located in front of the head, up front, so it's a carnivorous. The position of the eyes on the side of the head enable the prey to look for a wider region for looking out for the predator. Is it convincing?
Yes.
Okay.
I remember.
عايز اقول حاجه يا شباب لو الزوم disconnected you need to reconnect on the same link. Okay. Okay يا شباب.
اوكي
Okay.
We continue,
في نفس السؤال
we'll see another good example of nature selection. You can see a population of a varied insects, which have in figure 1, which have different colors, either dark or light. And due to the elimination of the lighter color, because it's obvious for the predator, so the population loses all the light colors by got eaten from the predator. Soon after that, the population which will be existing in the nature will be only of dark color. And this means that nature selected which species can survive, because eventually the insects with the light color body will eventually got extinct. So Darwin proposed, sorry? So Darwin proposed this natural selection as a mechanism of evolutionary adaptation of population to their environment. And the above example is a very good, and the above scenario is a very good example. So you can see a population of organisms have a combined between healthy variation that it got from parents and the production of offspring. So a lot of offspring are produced. Due to the environmental factor, in this example, the predation by the bird, this leads to difference in reproductive success, because the ones which is a dark skinned, dark covering will survive, leading to its adaptation and surviving in the population. A good example as well of that is the evolutionary adaptation of the bat. Over many years, these bats develop wings, and basically it's the only mammal capable of active flight. And this development in these characteristics happened over a lot of years and got refined over time by natural selection. So you can see here a comparison. By the way, this is my photo. You can see here, this is a comparison between the different apes to human. You can notice here, we have only 1.2% difference in our DNA between the Pompo chimpanzee, between the chimpanzee and the Pompo chimpanzee. So this actually 1.2 difference in our genetic material, that's enable us to behave like humans. And it's very, it's a surprising, and only 1.2% between us and chimpanzees. And of course, these evolutions have done over millions of years ago. So this part will conclude by two famous quotes, that biology is the most powerful technology ever created. DNA actually is the software, while protein are the hardware. And of course, cells are the factors for Gupta. While Schiffer have said that although nature needs thousands or millions of years to create a new species, man needs only a few dozen of years to destroy one. Okay, if you understand what I mean, that man can destroy a lot of habitat for looking for something he needs. So we'll talk now about levels of organization. So starting from the cells, you have some specialized cells. What's the meaning of specialization of cells? Some cells do one particular job, so they develop a distinct shape and do special kind of chemical reaction. This kind of thing, when these cells are grouped, there is a division of labor. So actually there is a specialization of the cells to carry out a particular function in an organism. So you can see as well, each cell contain a mitochondria, ribosomes, and other cell organisms which have another division of labor within the cell. The first example of a specialized cell is a ciliated cell. You can see from the figure, a cell that has cilia is bound as well with, in this figure as well, there's a goblet cell. And these two cells, the ciliated and the goblet cell, are characteristic for secreting. The goblet cell secrete mucus, while the cilia beats around, flicking continuously to move this mucus secreted from the goblet cell. And these kind of cells basically present in the trachea and our respiratory tract to sweep away bacteria. While the other type of specialized cells in our body is the nerve cell, which function in conducting the electrical impulses along the nerve fibers, and have a long fiber called axon where the impulses travel. This axon is surrounded and covered by myelin sheath. Actually, it's the fatty to insulate the nerve for the propagation of the nerve impulse or the transmission of the impulses. These nerve fibers are very long, starts from the central nervous system, which is the brain and the spinal cord, and to the body until reaching the extremes of the body. The chemical reactions which occur along these fibers and the nerve cell propagate through the nerve fibers for the impulse. The next type of specialized cells, of course, is the red blood cells, which function in the transport of oxygen through the binding of the hemoglobin which exists inside the goblet cells. The adaptation for this is very characteristics. You can see that it have a very important pigment called hemoglobin that when bind to oxygen, it's converted to oxyhemoglobin in lungs, and this binding is unstable. And of course, it needs to be unstable to be easily released to the tissues. These cells is got adapted by the absence of the nucleus to leave more space for hemoglobin for binding to oxygen. And these cells as well have a biconcave disc shape to increase the diffusion surface, and it's a very small, this type of cells, to enable the flexible squeezing inside fine capillaries. The next type of specialized cells is the egg cells or the ova. And this ova actually is larger than the sperm cells, and they are spherical. Their nucleus, of course, are haploid, and they carry the genetic material. What's meaning of haploid cell? Haploid, that's mean the ovum or the egg cells contain the half of the genetic material, because it needs the other half of genetic material from the sperm. Of course, it functions in reproduction and they develop into a baby when fertilized. So what about its adaptation? It has a nucleus with 23 chromosomes, because you know that the total number of chromosomes we have in each cell we have is 46 chromosomes. And that's why the egg cell is haploid, have the half number of chromosomes. While if it's have the 46 chromosomes, it's called diploid. Of course, it have these cells have a large amount of cytoplasm, which contain a yolk material or yolk droplets, which is made up of proteins and fats. And these yolk droplets or yolk materials, actually, it is a nutrient material for the growing embryo in future. This cell is covered by a jelly coat, mainly for preventing of the entry of more than one sperm. The next specialized cell is the sperm cell, which is actually haploid as well and carries the genetic material. The same, its function for reproduction and achieved by fertilization with the egg cell. Its adaptation, same like the egg cell, have a haploid number of chromosome, which is 23 chromosome. As well, it have an acrosome at the anterior tip of the sperm, which secrete enzymes that enable the sperm to lyse or digest the wall of the egg and the egg membranes for penetration. For its movement, its middle piece, as you can see from the figure, its middle piece is occupied with a lot of number of mitochondria, which provide the energy for movement. And it has a very long tail that enables the sperm to swim. You can see here a summary for the 5 different types of specialized cell, where it's found and the function, for summarizing the specialized types of cells. Continuing after cells, you have tissue. Tissue actually, it's a group of cells with similar structure, working together to perform a shared function. Example of that, the bone, the nerve, or the muscle tissue. You can see that cells can form an epithelium, like the lining of the mouth cavity. Cells can form a tube, like the kidney tubes. And you can see that cells can form like a gland, like a salivary gland. So here the cell combine together to form a tissue. The tissue, when they combine together, they form organ. So the organ is a structure made up of a group of tissue working together to perform a specific function. And of course, example of that is heart, lungs, intestine, brain, and eyes in animals. While the organ system actually is collectively, it's a group of organs that collectively form a system. And the example of that is, you can see that the circulatory system, it consists of a lot of other organs like heart and blood vessels, while the brain, spinal cord, and nerves make up the nervous system in animals. Eventually, the different organ systems will lead to an organism. The organism actually is formed of all these systems working together to produce an independent plant or independent animals. So you can see here, it starts with at the chemical levels, which is the atoms and the molecules, start to form a cellular level, which is like here the example of a smooth muscle cell, that when these muscle cells combined or combine together, they will form a tissue level. You can see the smooth muscle tissue, and this smooth muscle tissue together with another tissue, it form an organ level, and this organ is the stomach. Together, the organ with the liver and the pancreas and the gallbladder, small and a lot of other organ will form the organ system levels that eventually will lead to the organismal level. So here we'll talk about how we can see the cells. We can see the cells by using majorly two types of microscope. It's not only these two types, there is a lot of other sophisticated microscopes, but we will only deal with two types of microscope, the light microscope and the electron microscope. So you can see here, this actually the normal light microscope that you can see in the labs, even in the school labs or the university labs for seeing cells. That's a universal microscope which have all these structure. The light microscope, if you use a hand lens, it magnifies about 10 times, while the light microscope have two convex lenses and they can provide magnification up to 1500s, like the school laboratories, university laboratories can magnify to 400, magnification to 400. You have an eyepiece lens, which is actually 10x. So you can see the eyepiece lens is above here, this eyepiece lens magnifies around 10 times. The objective lens, you have three objective lenses. The largest one magnifies into 40 times. And you can see from the figure, here is the objective lenses. So the basic source of this microscope is the light, and it is provided either by a mirror or a bulb. You have two modes of adjustment, either coarse adjustment and fine adjustment knobs are used to sharpen the image. So you can see here from the figure, you have here the focusing knobs. You can see that's a coarse one if you want to focus, because every one of us has a different strengths of eyesight, and you have the fine adjustment for fine details. If you need to see fine details in the sample, you need to focus that. While the electron microscope magnifies about 10 times, a million times. You can use on this light microscope temporary slides. You can do temporary slides in the lab, but in the case of the temporary slides, the specimen can be dry out quickly, so they cannot be stored successfully. So usually we use a cover slip over the specimen, so to help to keep the specimen in place, and this slows down dehydration and protect the objective lens from moisture or stains. Another type of slide is the permanent slides. Usually we use a fixing material for the sample on the slide, and we use a resin like a glue called the Canada balsam to fix this cover over the slide, and this type of slide can be kept for many years. So if you look at these conversions, you can, of course, you have taken that before. We need to understand the conversion from centimeter to a millimeter to micrometer to a nanometer. Actually, 1 nanometer is a 10 to the power minus 9 meter. So in this figure, you can see the ranges of different structure to see the spectrum. So you can see that our eye can see up till 1 millimeter difference between two samples. So we can see up till the frog egg. But if you go deeper and you wanted to see a cell like plant cell or animal cell or some of the overall structure of bacteria or mitochondria, you need a light microscope for that. If you want to go deeper to see organelles and the smallest structure like viruses and ribosomes, you need to use an electron microscope. Of course, up till now, nobody has seen the atoms. There are some attempts to see the atoms, but there is no clear picture for the atoms. So here you can see the types of the light microscope. You have a bright field microscope. It is used for unstained specimen. It doesn't need to be a live specimen. And this uses light to see this specimen for identification. And this figure, it is some of the cheek epithelial cells are taken and and overlaid on a slide. And this bright field microscope, of course, the specimen is not stained. But if we try to stain the specimen, usually the specimen is stained, and you can see that you can use the same bright field light microscope to see more clearly the light specimen. In both cases, the specimen is not alive, okay? And in the other case, if you need to stain the cells, it needs to be fixed or preserved. So thereby you are actually killing the cell, and this type of slide can stay for longer, while the first type of microscope, if it's unstained, not fixed specimen, it will not got stored for a longer time. In this type of light microscope, it's called the phase contrast microscope. And in this type of phase contrast microscope, you actually can see viable cells. And the technology lies behind the phase contrast. Actually, the light passes and uses variation of density between structure in the cell to enhance the difference. And you can see that the figure is more sharper than the figure from the unstained specimen. And usually we use this phase contrast microscope in seeing living cells. The last type, which is a more sophisticated type of microscope, it's called the fluorescence light microscope. And in this type of microscope, we stain every structure inside the cell with a different fluorescent substance. This fluorescent substance, once you expose this specimen to UV, it will emit light in the visible range, so you can see it by eye. So you can see here that this specimen is stained by 3 different fluorescents. Actually, the blue one represents the nucleus, while the orange one represents the mitochondria, while the green one represents the cytoskeleton of these cells. And of course, this type of preparation of the specimen is done in non-living cells. So it's a very good way to understand how any abnormalities occur inside the cell by marking each organelle inside the cell by a different fluorescence. Here, the only difference between these fluorescence microscope and the other bright field and phase
repeat, the orange one refers to what?
Is the use of a UV as a main source of illumination.
Oh, thank you.
Sorry?
Thank you.
You're welcome. For the electron microscope, which can magnify up till 10 million times, you can see majorly there are 2 types of electron microscope, either scanning electron microscope or transmission electron microscope. We normally say SEM or TEM. So what about scanning electron microscope? Actually, this type of microscope magnifies, as I said before, up till 10 million times. So you can see here, we magnify the cilia. If you remember, the cilia of the specialized cells we have seen, the first type of specialized cell. By light microscope, you can see these cilia. But by scanning microscope, you can see that how these cilia appear very clearly. And usually the photos or the figures of the scanning microscope is black and white, and they use a special software to colorize this photo. So actually the scanning basically looks to the overall structure from outside, while transmission electron microscope, it looks internally into the internal structure of the cells or the tissue. So as you see, it is actually the same picture of the cilia, but this one, the specimen have been cut as a cross section or longitudinal section of the cilium to see the internal structure. So basically the major difference between these 2 types of electron microscope is how we look to this specimen. For scanning, we look from the outside to see the 3D structure or the 3D image, while the transmission electron microscope will look internally to see the internal structure. So here is a good example how we can use a microscope to calculate magnification. So if a specimen is used using the eyepiece, which is 10 times, with a lens, objective lens that is 40 times, the total magnification will be the multiplication 10 times 40, which is, so the magnification here will be 400 times. Organelles in cells are too small. The organelles are structure that is inside the cells are too small to be measured in millimeters. So we used a smaller unit, which is micrometer, which actually is, it's a 10 to power minus 6 meter. So each 1 micrometer is 10 minus 6 meter. So how we can calculate this? How we can calculate the magnification? The law says that magnification equals size of image over size of object. So you can see this structure as a photo. If you measure by a ruler, you'll find that it measures 35 millimeter. So if you reflect that on the equation, so you'll have magnification size of the image, which is 35 millimeter, over the size of the object, which is usually sometimes is given, it's a 10 micrometer, and you need to have both the numerator and denominator both have the same unit. So you need to convert the 35 millimeter into micrometer by multiplication to 1000. Soon you will find that the magnification in this figure will be 3500. So this is how we calculate the magnification using the law. In other case, you can measure the size of the object if you know the others parameter of the equation. So how we can know the size, the actual size of the object, that's the actual size, okay? But you need to be given the other unknown. So here the magnification is times 100,000. So the size of the object will be the size of the image. You measure it with the ruler, it's around 20 millimeter. The size of the object, the size of the image, as you said, is 20 millimeter, and the magnification is already given, which is 100,000. And when you do the math and making the unit equal, you'll find that the size of the real object will be around 0.2 micrometer. Here, a simplification of the law, you can see I equal M. I actually is the size of the image equal actual size times magnification. So in this first example, we calculated the magnification, and the size of the image is, you can calculate it again from using the ruler, and the size of the object was already given here. So the only unknown here was the magnification. While here we need to calculate the size of the object, the magnification is already given in the example. So, and there's the size of the image, you can get it by the ruler, and you do the math and the working out, and you'll get the actual size inside the cell to be 0.2 micrometer. Here you can see this slide is like a working out exercise. You know, you need to do it at home and submit your answers on Moodle. Take your time, you will find the quiz on Moodle. There is exercise 1 and exercise 2 using the same law, the same way I used to calculate to submit your answers. So we'll talk now about homeostasis. So homeostasis can be defined as how our body regulate its internal environment to keep a constant state of balance between its different physiological function. The perfect example here about regulating the body temperature. So you can see two types of organism here. A river otter, which is a temperature regulator, and a large mouth bass, it's a kind of fish, which is a conformer or adapter. You can see that as long as the ambient or environment temperature increases, the fish increases its body temperature to got adapted with the external environment, while the otter do a lot of internal mechanism to preserve its body temperature around 37 degrees Celsius. So you can actually, basically, you can see two different organisms. One of them can regulate its body temperature, but the other one is an adapter to the external environment. The adapter or the conformer here is a fish, while the regulator is the river otter. So you can see here, fish actually it's a cold-blooded animals, while the hummingbird is الناس اللي لسه فاتحه دلوقتي who just opened the presentation, they can go for the recorded lectures after the presentation. For the hummingbird, you can see that it is an endothermic, warm-blooded animal. So actually, it is a regulator. And for as well, the ectothermic animal like the iguana, you can see it's an ectothermic, cold-blooded animal. So in the so here you can see this one, the otter, actually it's an endothermic, while the bass or the fish is an ectothermic. Why ectothermic? Because it depends to the external environment on maintaining its body temperature. While the first one is an endothermic, it doesn't rely on the external environment. So to keep this homeostasis occur, bodies have two basic mechanisms: the negative feedback mechanism and the positive feedback mechanism. This negative feedback mechanism counteract the effect of changes in the internal environment and responsible for maintaining homeostasis, while the positive feedback mechanism creates cycles and to amplify the signal inside our body. For both these systems are used for regulating homeostasis. You can see here an iguana actually iguana is a cold-blooded animal. It's an ectothermic animal. So it increases and decreases its body temperature based on the environment. And you can see how the iguana can control its body temperature based on 4 factors, either by using radiation. And radiation, actually, it's the waves coming from the sun. The iguana absorbs some of this heat from the sun if the iguana needs to increase its body temperature, and radiate away some of this energy to the surrounding environment. What about if this iguana got overheated? What it needs to do? It does some of the evaporation, like the removal of heat. The evaporation, it removes heat by evaporating water from its nose and mouth to its external environment. This leads to a strong cooling effect. As well, convection, you can see that convection actually it is the transfer of heat by movement of air over the surface. So by standing this iguana, the air can move over its dry skin when the blood moves in the skin and leads to evaporation of heat outside of the body. As well, it can gain heat from conduction over a rock. So if the rock either cold or hot, this heat, if it's hot, the heat will be conducted inside, conducted to the liver to elevate its heat. So actually, these 4 mechanisms the iguana is using for doing homeostasis and regulating its body temperature, or adapting its body temperature to the external environment by doing this for a different mechanism. The perfect example in our body is controlling the thermoregulation, how body respond to changes in our temperature. So the normal body temperature is around from 36 to 38 degrees Celsius. When body temperature increased, there is a thermostat inside our pituitary gland, which is the hypothalamus, activate this cooling mechanism. This cooling mechanism lead to 2 main effects. Blood vessels in the skin dilates. Blood vessels dilates. This blood vessels is full with warm blood, so heat radiates out from the skin surface. Another effect is the sweat gland start to secrete sweat, which evaporates, cooling the body, and then the body temperature decrease. While in the other case, when the body temperature decreasing below 36, there is two other different mechanisms controlled as well by the thermostat in the hypothalamus in the brain. This leads to two other major effects. The opposing effect of the dilating of the vessels, blood vessels, actually it is constricting the blood vessels. So once there is a constriction of the blood vessels, the blood will not be near the surface of the skin, so that the heat will not be lost to the environment. And as well, the muscles start to contract vigorously and rapidly, causing shivering and generate heat. This raises the body temperature until reaching from 36 to 38. So actually we have a negative and the positive feedback, that one of them decreases the effect of change and one of them enhances the effect of change. Because positive feedback effect can be spiral out of control. So actually we find that this positive feedback effect is relatively rare in biological system. Most of the mechanisms that control homeostasis in our body and other animals, most of them are negatively feedback. The best example of positive feedback during labor, when we can see that in the figure. You can see when the baby starts to push in the cervix, so that's an example of a positive feedback. When the baby starts to push against the cervix, it is causing the cervix to stretch. This stretching of cervix causes nerve impulse to be sent to the brain. This brain stimulates the posterior pituitary to release an important hormone called oxytocin. This oxytocin causes the smooth muscle to contract in the lining of the uterus. This leads to the delivery of the baby. The continuous cycle of this leading to the overproduction of oxytocin, to have more contraction. So actually that's an example of a positive feedback mechanism that enhances the effect. But as we said before, these positive feedback are rare in the animal. So how we can see the organization of our body? As we said before, cells are organized into tissue, group of cells with similar appearance and common function in the tissue. There are 4 types of tissue, and today we will deal only with the epithelial tissue, which is the main covering tissue in our body. Different types of tissue are further organized into function units, the organs. The simplest animal is the sponge. You can see it in the sea. It lacks organs or even through tissues. The group of organs form the organ system. As you can see from the figure, how epithelial cells, when combined with the different other cells, will form tissues, and then will lead to the organ. In this case, is the bladder that soon will form the urinary system. As we see from the figure, the urinary system consists of urethra, bladder, ureter, and kidney. So here you can see for the epithelial tissue, the main characteristics of the epithelial tissue is it lines, covers, protects other tissues and organs. It is tightly junctioned together and have a free surface. Actually, the different type of epithelial tissue is named according to a cell shape. This tissue function is as a barrier against mechanical injury, pathogen, and fluid loss. It is thin and lacks, it is the only tissue that lacks blood vessels. It can form an active interface with the environment. So this epithelial tissue can form active interface with the environment. Like the epithelium lining the nasal passages, which is crucial for the sense of the sensation. أيوة محمد.
دكتور بس في واحدة اسمها علا منصور راح عندها الإنترنت بس.
It's okay. She can reconnect and see the lecture, the recorded lecture after that. Okay. So the types of the tissue, you can see the first type is called the stratified squamous epithelium. So it's a multi-layered epithelium and can regenerate rapidly. And the new cells are coming from the basal surface, and the new cells are pushed upward, replacing cells that is damaged. And this type of stratified squamous is found in the skin in the lining of the mouth, in the lining of the anus, and in vagina. All of these regions characterized by continuous friction of the material passing through it, so it needs to be regenerated. That's why it's a multi-layer. For the other type, the cuboidal epithelium, you can see that it is a disc-shaped cell and only specialized for secretion. And it makes up the epithelium of kidney and many glands, including the thyroid gland and salivary glands. While the simple columnar epithelia, you can see that the nucleus is located below, and you have a tall cell, large brick-shaped cells, and as well it's located at the region of active secretion and active absorption. And this type of cell exists in intestine, lining the intestine, secreting digestive juices and absorbing nutrients. While the other type is a simple squamous epithelia, which is a plate-like cells, actually mainly function of material exchange by diffusion. These 10 leaky lines blood vessels, so it's a major lining of blood vessels, and the air sacs of the lungs. And actually, this is convincing, because in this region there is always exchange between the inside the blood vessel and the outside of the blood vessel, either nutrient and gases. So these blood vessels and the air sacs has to be lined by these simple squamous epithelium. And the name simple implies that this type of epithelium consists only of one layer. In the last type of epithelium, it's called the pseudostratified epithelium. What's the meaning of stratified? Stratified, that means it's many layer, a multiple layer. But why here it's a pseudo? Pseudo because it's a false stratification. Because you can see that it appears like it has two layers above each other, but they are only one layer, because not all of the cell reaching the border above the cells. So it is a pseudo stratification. And this single layer of cells is varying. That's why it's a varying of height and position. And this ciliated type of cells, the one we said at the beginning of the lecture, it secretes mucus by the help of the goblet cell, and usually present in the respiratory tract to sweep the film of mucus outside our trachea and the respiratory tract, which sometimes full of pathogens and bacteria. You need to understand a very important thing, that cells have a polarity of epithelium. What's the meaning of polarity? That each cell, especially the columnar cell, have an apical surface and they have a basal surface. This polarity, that means the cells have two different sides. The apical surface is facing the lumen, the cavity, or outside the organ or the tissue, and that's why it is exposed to the fluid or air. This apical surface, as an example of the lining of the smallest intestine, is covered by a lot of some structure called the microvilli. This microvilli increases the surface area for absorbing nutrients, while the opposite side, which is the basal surface, is bound to the underlying tissue. So you can see here a more clear picture between the simple squamous, and you can notice if you look from above to the simple squamous, if you look from above, it looks like flat cells, and you can actually see the nucleus. It's as well, you can see here the simple cuboidal, it's a cube-shaped cells, the nucleus inside the center of the cell, and from above it appears like a square shape or cubical shape cells. While the pseudostratified, you can see that not all the cell reaches the border, reaches the luminal surface or the apical surface. That's why it is a pseudostratified. And this is a real image of a pseudostratified epithelium. And you can see that the border of cilia is above, and you can see the mucus, and it's actually present in the respiratory tract. Here you can see the pseudostratified columnar epithelia. You can see here, the pseudostratified epithelia, as the stratified squamous epithelia. You can see the difference. And actually, this is a perfect example of our skin. You can see that the outer surface of this type of epithelia, usually dead cells. and this dead cell got swept away by friction and have to be regenerated from the basal cells. Okay, so this is the major types of epithelium. You can see here different photos for epithelia. And now I finished my lecture. So you need to do now, you go for Moodle. The Moodle quiz will start at 11:45. At 11:45, you will have around 5 minutes to answer the quiz. We'll meet after the quiz on the same link of Zoom for discussion. Good luck يا شباب. 11:45